Hello, welcome to our 7th AP Biology lesson. Today our lesson will be on new material in Unit 7. On Friday, April 3rd, an announcement will be made providing details about the 2020 AP Biology exam. Beginning on April 7th, we will be presenting lessons that will review the first 6 units in the AP Biology course. All lessons, including these with new material, will include practice with science skills, which are relevant with any content. We will also provide practice with free response questions in each lesson. My name is Allison Kate. I'm affiliated both with UC Berkeley and Redwood High School, and I'm teaching today from my home in El Cerrito, California. So let's get started with our warm-up today. If you're just joining us, the warm-up is a question from the previous lesson. It's given for homework at the end of the previous lesson, which yesterday was lesson 7.8. And in that lesson, one of the content topics was about the endosymbiotic theory. So this question is about that. If you are just joining us, go ahead and grab a pen and paper and see if you can answer it as I debrief it. And if you did this last night for homework, good for you. And go ahead and get that out as I go over the scoring guidelines. All right, so let's read the question together. According to the endosymbiotic theory, some organelles are believed to have evolved through a symbiotic relationship between eukaryotic and prokaryotic cells. Describe 3 observations that support the endosymbiotic theory. Okay, so you're asked for 3, and you're asked to use that command term or task verb, describe. So what you're looking at on the scoring guidelines is a lot of bullet points. The ones on the left were the bullet points that were most often used to answer this question. The ones on the right also appeared, but least often or less often. You need to come up with 3 descriptions, and so each bullet point would have earned you 1 point. Remember, we're trying to provide evidence for endosymbiosis. So either mitochondria or chloroplasts containing their own DNA, like prokaryotes do, would have earned a point. Mitochondria and chloroplasts can both self-replicate, like prokaryotes. Either one of those would have earned you a point. And mitochondrial chromosomes and chloroplast chromosomes are circular, like prokaryotic chromosomes. Finally, one way most often response was that mitochondrial chromosomes lack histones, as do prokaryotic chromosomes. And then I'm not going to read through the other long list, but you can take a look at those and see if you came up with any of those. So that was a 3-point question. I hope you did really well on that one. All right, so today's lesson overview. Today we will be introducing you to the concept of phylogeny and to science practice skill 2DC, which if you don't have your CED in front of you, deals with visual representations. And so when we look at phylogeny, we're going to see that both phylogenetic trees and cladograms are models of the history of a species that represent this visually. So we're going to look at diagrams of both. We're also going to look at sample multiple choice and free response practice questions that combine the content of phylogeny and the skill of explaining relationships in models that are represented visually. You should already be familiar with the concepts of species and speciation and the terms phylogenetic tree and cladogram. And if you haven't been exposed to those before, you will hear them a lot in this lesson today. By the end of this lesson, you'll be able to describe the types of evidence that can be used to infer an evolutionary relationship. You'll be able to explain how a phylogenetic tree and or cladogram can be used to infer evolutionary relatedness, and you will be able to explain the relationship between different characteristics of biological concepts, processes, or models that are represented visually. So don't forget your pen and paper, and let's get started. I want to tell you where we are today in terms of the overall framework of the AP Biology course. Again, today we're working on topic 7.9, which is in the natural selection unit. We're working on the idea of phylogeny. And there are two learning objectives, which I read to you out loud in the previous slide. You can see there. And our essential knowledge for today's lesson is that phylogenetic trees and cladograms show evolutionary relationships among lineages. So today our topic skill pair is phylogeny and explain relationships that are represented visually. So here's what you need to know. First of all, what is phylogeny? Phylogeny is the evolutionary history of a species. And the way that we represent those relationships visually is either using a phylogenetic tree or a cladogram. And we'll talk about the differences between those. So phylogenetic trees will show the amount of change over time that is calibrated by both fossil evidence or a molecular clock. And if you look at the phylogenetic tree image, you can see that we have a history of life. We get the three domains, bacteria, archaea, and eukarya, and it shows kind of that common ancestor at the root, at the bottom of that tree. Maybe I can point that out with this pointer. Okay, and then the cladogram is on the right. It's kind of a blurry image, but I'm going to be showing you a bunch more cladograms as we proceed through this lesson. So phylogeny, there are some important vocabulary words or concepts that you should understand when we are going to be constructing these diagrams. And so you can use traits that are either gained or lost during evolution to construct these phylogenetic trees and cladograms. But let's take a look at some of the important ideas here. First of all, shared characteristics are present in more than one lineage. Shared derived characteristics indicate common ancestry and are informative for the construction of phylogenetic trees and cladograms. And then finally, the out group represents the lineage that is least closely related to the remainder of the organisms in that phylogenetic tree or cladogram. And I'm going to give you some practice so you can see what that outgroup looks like. You'll see it a couple of times during today's lesson. A few other points about phylogeny and the type of data that can be used to construct these visual representations. Molecular data typically provide more accurate and reliable evidence than morphological traits, that is how an organism looks, when constructing these trees and cladograms. Phylogenetic trees and cladograms can be used to illustrate speciation, whether it has occurred or not. The nodes are a really important concept. The nodes on a tree represent the most recent common ancestor of any two groups or lineages. And I'll be pointing some of those out to you as we do our practice. Phylogenetic trees and cladograms can be constructed from morphological similarities of living or fossil species and from DNA and protein sequence similarities. And in just a moment, I'm going to take you through a lab that you might do if you were in class right now that will have you start off with morphological characteristics and then refine it using molecular evidence. In this case, it'll be DNA sequence evidence. And finally, phylogenetic trees and cladograms represent hypotheses and are constantly being revised based on evidence. So the lab that I take you through will also give you practice at revising your original hypothesis. So the lab that I'm talking about is from the College Board AP Biology Lab Manual, which you can access on AP Central. This is also a lab that if you have access to a computer, you can do on your own. I'm going to be providing you with some screenshots so that you can work on this after you watch this lesson and give it a try because it's kind of fun. So this is AP Biology Investigation 3, and it uses a tool called BLAST, and I'll be explaining what BLAST is in a minute, but it allows you to compare DNA sequences to understand evolutionary relationships. So BLAST is short for Basic Local Alignment Search Tool. It's kind of a mouthful, so we just call it BLAST. BLAST is a bioinformatics tool that allows us to search a very large molecular database quickly to determine evolutionary relationships that are based on either DNA or protein similarities. In this particular lab, the objective is to determine the organism that is most closely related to one discovered in an unknown fossil. So this lab starts off with you being a paleontologist, and you go out and you find this fossil. And the first thing that you're asked to do is to construct a cladogram that represents your hypothesis based on morphological characteristics alone. So if I were back in my classroom, I would take this image that you see in figure 3 of the fossil that you guys just collected out in the field, and I would project it, and I would ask the students to see if they can identify any morphological characteristics. So I know it's kind of small, but if you look closely, you can see that there are some limbs. I see 2 for sure, maybe 3, maybe 4. It's a little hard to tell. I can see that this organism has a tail, and I can see that it has some backbones or vertebrae, maybe even some ribs. Your eyes are probably better than mine. And I can also see that there is an opening in the skull. So you can take a look at this and see how many characteristics you can come up with. And then the first thing that you're going to do, and we'll do that together in the next slide, is to place this organism on a cladogram based on those traits alone. Then I'm going to teach you how to use the BLAST tool, and I'm just going to have to do that through a series of screenshots. And then finally, you're going to be given 4 gene sequences that come from this fossil, and you're going to use those to compare the fossil genes to extant or living gene sequences. And then finally, you'll use that information to revise your cladogram in light of the molecular evidence that you get using this BLAST tool. So the first thing we want to do is indicate where on this cladogram we might hypothesize this organism should be placed. So we know that it has vertebrae. So we know that we're going to place it above this node because that would be the last common ancestor. Some of you may, based on how it looks, may want to travel up and take a look and see maybe it belongs in the bird lineage or the crocodilian lineage. We don't have any evidence of fur, but it would be unlikely that that would be preserved in fossil form anyway. So, I mean, we're kind of thinking that it was going to be in that area. Okay, so that's your working hypothesis based on the traits that you can observe or see. So now we're going to use the BLAST tool. Again, that's the basic local alignment search tool. We're going to see how well our fossil gene sequences align to living gene sequences. And so this is the home page of this tool. And you can Google the NCBI GenBank and or BLAST, and it will come up. We are going to be using the nucleotide BLAST, but you could also do a protein BLAST, which you can see on the lower right side of your screen. So what you're going to be doing is beginning with one of the fossil gene sequences, and you're going to upload them one at a time. So when you do that, you're going to see your job title, and that's your AP Biology fossil specimen gene 1. You'll upload it into this box, and what you'll see is a series of A's, C's, T's, and G's. That's your gene sequence from this fossil. And then you're going to look down here, and you're going to choose which database you want to search. If you only wanted to search the human genome database, you would click that button. If you wanted to know mouse, we want to know all of them. This is a drop-down menu that you can also optimize, and we want to look at gene sequences, not these RNA sequences. So we would include that as well. So that's getting set up. And then the last thing we want to do is we want to optimize for highly similar sequences. We're going to get a lot of hits. We want the hits that are most closely related to the sequence that we put into the tool. And then you hit blast, this button here, so you blast off. And then what happens is literally in a minute or two, this database is searched for those sequences that most closely match the one that you put in. So you might see results that look like this. And again, it just looks like a gene sequence. So what the heck does that mean? So you can look at a little bit more information about that gene sequence. You can see that it comes from an organism with this scientific name coding for this protein. So this is collagen, and it codes for one of the chains. And we did indicate more from the morphological traits that we saw some vertebrae or bones, limbs, other bones, and collagen would be an important component, an important protein for an organism that has bones. If you don't know what the heck this scientific name is, you can come down here and you can see that this is in fact a turkey. That's the common name of this organism. And then you can get the full, you know, classification for this turkey. And then if you think about it, when we go back to that cladogram that we hypothesized, we put it after the vertebrae. We weren't sure if it was going to be on the bird lineages or the crocodile lineage, but we were pretty close with our hypothesis. Now we have some more evidence. This is molecular evidence that it is probably related to a bird. Then you would go ahead and you would upload the other 3 genes and see what information they provide. The other thing that's pretty cool about this tool is that it gives you a cladogram. They call it a distance tree of results. And so you would click on that, and what you would see is something like this. And you can see that there's a lot of information here, and you can see going back to the nearest common ancestor. Let's enlarge this, and your gene of interest is highlighted. And then you can see here that this is most closely related to a Gallus-Gallus collagen, the code for Gallus-Gallus collagen. And if you're not sure what Gallus-Gallus is, you can see that it's color-coded. So you can look at the color coding. And again, that turns out to be a chicken, and a chicken is a bird. And so we know that we're on the right track placing that organism in that area. So then you'd go back and you would refine your hypothesis based on this new molecular evidence. Okay, so if you want, go ahead and try this on your own. It should be fun. All right, so some key takeaways. Phylogeny is the evolutionary history of a species. Both phylogenetic trees and cladograms are visual representation that shows these evolutionary relationships. The difference is that phylogenetic trees show the amount of change over time, and that's calibrated using fossil evidence or molecular clocks. And finally, molecular data typically provide more accurate and reliable evidence than morphological traits in the construction of phylogenetic trees and cladograms. And we're going to investigate that coming up and why that might be. So let's get some practice. My first practice is showing you a phylogenetic tree. And this figure shows relationships of organisms based on catalase structure. Catalase is a protein enzyme that catalyzes the breakdown of hydrogen peroxide, which is H2O2, into water and oxygen gas in the cells of many living organisms. And you may have worked with this in your enzyme lab when you did your enzyme lab in class. Students used an online database which used the structure of catalase from different species to generate the phylogenetic tree shown here. Okay, so let's look at what the question is asking us to do. It's asking us which of the following student claims is best supported by the data provided. Okay, so the first claim is that mushrooms and potatoes are most closely related since the node they share gave rise to all of the other organisms. So now you need to look at mushrooms and potatoes, the node they share. Did it give rise to all of the other organisms? Is that the data that is going to support that claim? Cattle, which we see here, are equally related to pigs and sheep because phylogeny places the cattle between pigs and sheep. C, yeast and mushrooms are most closely related because they are both fungi. And finally, D, cattle and sheep are separated by only one node, and they are separated from the other organisms by more than one node. So this is the node. The cattle and sheep are separated by one, and the others are separated by more than one. Okay, so do you have your answer? I hope you chose D. D is the correct response. Cattle and sheep are separated by only one node, and they are separated from the others by more than one node. All right, let's try one more multiple choice. Now we're looking at a phylogenetic tree of some island birds, and you may want to think back to an earlier lesson about Darwin's finches. And when we're talking about the morphology in this case, we're talking about the beak. How does the beak look? And you can see that it's different in each one of these species. So a group of birds is found in an island chain that have all descended from a single ancestral species, and that would be here. Okay, so this is the common ancestor of all 10 species. And the question is, which of the following best explains the divergence in morphology in the birds? In other words, why do they all have different beaks? Okay. So here are the choices. A, the original habitat contained mutagens, leading to the rise of new species as a result of accumulated mutations. B, the original habitat had many available unoccupied ecological niches that were colonized by populations of the original species. C, the original habitat contained many predators and the species diversified in order to avoid extinction. And D, the original habitat was tropical, which increased the rate of mutation and led to rapid diversification. So in this case, I want you to think about some of the things that we have studied in previous lessons. I want you to think about Lamarckian language. I want you to think about habitat and mutations. So the correct answer in this case is B. The original habitat had many available unoccupied ecological niches that were colonized by populations of the original species. So as the original species was able to occupy a different niche, they were able to exploit a different food source. And so adaptations that helped them obtain that food source would allow them to survive and reproduce. And over time, they would develop those different morphologies in their beak structure. So they'd look different. All right, let's take a look at a FRQ. And in this particular FRQ, we're only going to look at part B. And I know that in yesterday's lesson, Ms. Pfizer had you take a look at a data table that looked a lot like this. So hopefully you can apply that to this question. So again, the statement that phylogeny is the evolutionary history of a species kind of points us into what we're going to need to do. In part B, we are asked to do 4 different things. So look at those task verbs. The first thing, it's asking us to use the data in the table. So based on the data in the table below, draw a phylogenetic tree that reflects the evolutionary relationships of the organisms based on the differences in their cytochrome C amino acid sequences. So you are being asked to draw a visual representation of these 5 species based on the data in this data table. Then you're asked to explain the relationships of the organisms. In other words, why did you draw the tree the way you did? And then you're asked, based on the data again, identify which organism is most closely related to the chicken and explain why you selected that organism. So let's get into this data table, which is showing us the number of amino acid differences in cytochrome C among various organisms. So we're talking about the protein, cytochrome C, and we're looking at the amino acid differences in a horse, donkey, chicken, penguin, and snake. And so let's take a look at this row. So there's 1 amino acid difference between the donkey and the horse. So that tells you that they're probably pretty closely related to one another. There are 11 between the chicken and the horse and 10 between the chicken and the donkey. The penguin has 13 between the horse and the penguin, 12 between the donkey and the penguin, and then only 2 between the penguin and the chicken. Okay. And then finally, did I do that right? 2, 3. And then the snake has the greatest number of amino acid differences from the horse. Okay, so let's start with that information and look at how we could construct our phylogenetic tree. So the scoring guideline shows us a phylogenetic tree over here with a root showing its common ancestry, and then this shows it in that cladogram form. And again, the snake and the horse have the greatest number of amino acid differences, so they're going to be the ones that are least related. The donkey and the horse were the most closely related, so they're going to be next to each other on this tree. And then you can fill in based on the number of amino acid differences from the chart. So either one of these would have earned 1 point for correctly drawing the tree. The second part asks you to explain how you constructed that tree. And you could have said this either way. You could have said that the more amino acid differences, the less closely related they are. So the horse and the snake had 21. Or you could have said the fewer differences, the more closely related they are. So the horse and the donkey had 1. So they're most closely related. Clearly, the penguin is most closely related to the chicken, and because it had the fewest amino acid differences compared to the snake and the donkey and the horse. Okay, so that's a little practice at both drawing a visual representation and then kind of understanding how to use that data table showing you those amino acid differences. And again, that's molecular data, not morphological data. All right, so in this case, 2 of the task verbs were explain, so I wanted to just get some practice with that task verb. Explain means to provide information about how or why a relationship, process, pattern, position, situation, or outcome occurs. And it wants you to use evidence and or reasoning to support or qualify a claim. So we just did that on our last FRQ practice. If you get an explain how, it typically requires analyzing the relationship, so analysis. And if it's explain why, it requires analysis of motivations or reasons for the relationship, process, pattern, etc. So pay close attention to that task verb if it's just explain or explain how or explain why. So let's get some practice with that. This is a really long prompt. And then after this prompt, there's going to be a visual representation, a model, a phylogenetic tree that we're going to be asked to use to explain something. Okay, so I'm going to read this out loud, and I'd like you guys to read along with me so that when we get to that phylogenetic tree, you have your background. All right, so river dolphins are morphologically distinct from other dolphins and are primarily restricted to freshwater ecosystems in Asia and South America. Determining the phylogeny of these animals based on morphology has been difficult because they have a large number of derived characters, including a long, narrow rostrum, which is a scientific name for beak. They have a low dorsal fin, a flexible neck, and eyes that are greatly reduced. River dolphins were originally classified together based on their morphology, but molecular studies have shown they are less closely related than previously thought. Scientists analyzed the nucleotide sequences of mitochondrial genes, including the genes for cytochrome B and 2 ribosomal RNAs. By combining the nucleotide sequence data with analyses of embryonic development and morphology, the scientists have proposed the phylogenetic tree shown in Figure 1. And in this case, we're going to see that baleen whales, they're filter feeders, and they're going to be used as our outgroup in this phylogenetic tree. So you're going to get your example of an outgroup. All right, so here's the phylogenetic tree of cetaceans, which is the family that includes both toothed whales and baleen whales. And you can see that the baleen whales are an outgroup because they do not contain teeth, okay? So they feed differently. I also want you to see that we're mostly concerned with the river dolphins. So we've got a Southeast Asian river dolphin, Chinese river dolphin, Amazon river dolphin, and La Plata river dolphin. And you can see they're all in their own lineage. D, this is part D. This was a whole question, but we're only going to look at part D. It says that molecular data indicate that river dolphins do not form a monophyletic group. That means that we don't see them all in one lineage. Explain why these animals nevertheless have morphological similarities. And before we go over the answer, I want you to think about something that we talked about in a previous lesson. When we talked about lesson 7.3 on artificial selection, we talked about how humans select for certain traits, but that in convergent evolution, the environment selects the traits. And so that sometimes individuals who are not related, they have different evolutionary lineages, they can resemble one another due to the environmental pressure. So please keep that in mind as we go through our choices or our thought process. Okay, so this is a free response question that's asking you to explain why these animals look the same even though genetically they're different. And your answer should include some way of getting in that the similarities in morphology are likely due to convergent evolution. And the reason for that is that river dolphins inhabit similar habitats and are subject to the pressures of those environments. So reduced visibility in most large rivers occurs due to silt, and that would lead to a reduction in vision or eye size. Long noses are an adaptation for searching for food in that environment, and streamlined bodies and dorsal fins are not as critical for stability in rivers as they are for some of the other organisms in that tree that live in the ocean, like some of the other whales. So this kind of puts it all together, looking at convergent evolution and how that can be shown in a visual representation, such as a phylogenetic tree. All right, so just a reminder about task verbs. We're going to practice these in all of our lessons. Task verbs are not just in evolution. They're used through all units in the curriculum. You can find them in the course and exam description on page 199. And again, you can find that course and exam description on AP Central. And again, since you're not in the classroom and your teacher, don't have your teacher kind of guiding you through these things, I think it might be a good idea for you to look at those on your own. Okay, so let's do a couple of checks for understanding. So remember, the skill that we're practicing is to explain the relationships between different characteristics of biological concepts, processes, or models that are represented visually. So again, in this content, phylogeny, we're going to be talking about cladograms and phylogenetic trees. So what you're looking at is a cladogram of selected vertebrate groups. It looks a lot like the one that I showed you on the very first slide that was kind of blurry. And this question reads, whales, bony fish, and cartilaginous fish all have similar adaptations that enable them to live in the ocean. This figure shows the phylogenetic relationships of selected vertebrates. Your question is way down here. Based on the phylogenetic relationships shown in figure 1, which of the following best explains the similarities exhibited by whales, bony fish, and cartilaginous fish? So we're looking at whales, bony fish, and cartilaginous fish, and you want to kind of look at the lineage. Okay, so these nodes represent the last common ancestor. Okay. Okay, so your answer choice is A, convergent evolution, because these organisms do not share a recent common ancestor. Look for a shared recent common ancestor on that tree. Adaptive radiation, because all three groups evolved from a unique shared common ancestor from the cartilaginous fish group. Okay, is that true? C, mutation, because shared traits can be traced back to a recent common ancestor. And D, founder effect, because all three groups previously exist together as one population that was then disrupted, causing speciation. So look at your choices, think about what we've already talked about today, look at your phylogenetic tree or cladogram, select your choice. Okay, the correct answer is A, convergent evolution. These organisms do not share a recent common ancestor. So look at those nodes for the common ancestors and look at where that recent common ancestor would be, and you don't see it. All right, so let's debrief, and then we're going to get some practice with some more visual representations. So you should now be able to describe the types of evidence that can be used to infer evolutionary relationships, right? Morphological traits or molecular traits. You should be able to explain how a phylogenetic tree and or a cladogram can be used to infer evolutionary relatedness. You want to look for shared characteristics and shared derived traits. You want to look at the node that represents the most recent common ancestors. And finally, you should be able to explain between different characteristics of biological concepts, processes, or models that are represented visually. Your key takeaways. Evolution is characterized by a change in genetic makeup of a population over time and is supported by multiple lines of evidence. Phylogenetic trees and cladograms are both visual representations of evolutionary relationships. So let's take a look at a FRQ. I believe this was from last year's AP exam. And we can take a look at some common errors and misconceptions that student had in their responses on this question. So we're looking again at that data table, which you've had some practice with, and we're looking at molecular evidence. We're looking at the divergence. These are percents of mitochondrial DNA sequences among 5 primate species. So you can see we have humans, gorillas, orangutans, gibbons, and chimps. And again, if you kind of look across, you can see that we see the gibbon at 18.1% has the greatest difference from the human. Okay, the orangutan has 16.1, which is the next greatest difference from humans. You can look at the gorilla at 10.3%, which would be the next, and then the one that has the fewest differences in percent, 8.8% from the human, is the chimp. Okay, so you were given the template on the question, and they also provided an outgroup for you. So this outgroup is the gibbon, and it would be the one that is least related to the other ones. It's on its own lineage. And so looking at our task verbs, using the data in the table, construct a cladogram on the template provided, and then provide reasoning for the placement of gibbons as the outgroup on the cladogram. And then finally, it asked students to draw a circle around all the species indicated by the node in the square that are related. Okay, so think about how you would answer that based on these differences in the percent divergent in these primate species. May even have that pencil and paper and draw your cladogram. And then the final thing is you want to think about the organisms that are going to be related to the species that are shown by this node with the square around it. Okay, so from the top to bottom, the human, the chimp, the gorilla, the orangutan, and the gibbon. And the reason for that is that they are the human and the chimp have the least divergence, the human and the gorilla the next least, as I went over on the data table, and the human and the orangutan had the most on those ones, and then compared to the outgroup, it had the most. So the reasoning why the gibbon is the outgroup is that it is the least similar to all of the other species, or you could assess the one that's most different from all of the other species. Or you could have used the term divergent. It's the one that is most divergent from the other species. And students did pretty well with that. However, where the error and misconception came, so you were supposed to correctly circle the 3 species in the first 3 positions of the cladogram to show the ones that are descended from the species indicated by this node. And so it's all of the ones that are above that, which would be the gorilla, the chimp, and the human. And students had a difficult time doing that. So that is was a knowledge gap on this one. So here's your homework for tonight. This question will be the warm-up tomorrow, and my colleague, Ms. Pfizer, will go over this with you. It gives you more practice with the data table and more practice constructing a phylogenetic tree. So again, this is based on molecular data. The amino acid sequence of cytochrome C was determined for 5 species of vertebrates, and the table below shows the number of differences in the sequences between each pair of species. This time you're given some scientific names. It doesn't totally matter. In some ways, you know, it may be a little bit harder to not know them, but really what you're looking at is the difference, right? So you can see that these guys, it's 0 and 1, and these guys is 0 and 21. These guys is 0 and 11, and these guys is 0 and 13. So key in on that. And you are asked to, using that data, create a phylogenetic tree using this template. So you're just going to write those organisms where you think they belong here, and then provide reasonings for why you placed them on the tree, and why the placement on the tree of the species that is least related to the others. So if you're wanting to do this, you can kind of take a picture, do a screenshot, or this will be recorded and you come back to it later to do your homework. Okay. So one word from the College Board. We all know that not all students have access to the internet or a device. We're working on solutions to help students get what they need to show their best work. If you need mobile tools or connectivity or know someone who does, you can reach us directly to let us know and reach the College Board at cb.org/tech. And this will be especially important since you'll be taking the AP exam on your own device. So you want to get that as soon as possible so you get familiar with it before you have to take your exam. Okay, so again, thanks for watching. I hope you'll join us for all of our lessons. Make sure you complete that practice question for homework, and Ms. Pfizer will review it with you in tomorrow's lesson. Have a great rest of your day. Thanks for watching.